We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. Carta brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
Hi everybody, thank you for inviting me to give this presentation today. So today I'm going to give you an overview about Salmonella in humans and other animals. So the pathogen Salmonella are gram-negative bacteria. As you can see here, those are these gram-negative rods. The bacterium is motile due to Speritrichos flagella. It colonizes the gut of animals, and there are two species, Salmonella enterica and Salmonella bongorai. So you can see here that Salmonella enterica has 6 subspecies, and there is only Salmonella bongorai, and these Salmonella, they primarily colonize cold blooded animals like reptiles. Subspecies enterica 1, however, is where there are all the Salmonella strains that you find in humans. They can infect humans as well as other mammals, and these include Salmonella enteritidis and Typhimurium, as well as Salmonella typhi and paratyphi that are really specific to humans. Non-typhoidal Salmonella is one of the primary classification of Salmonella. So we have non-typhoidal and typhoidal Salmonella. And non-typhoidal Salmonella, there is over 2,000 serovars, but the most prevalent one are Salmonella typhimurium and Salmonella enteritidis. In a given year, we have at least 100 million cases worldwide, and antimicrobial resistance is an emerging issue. And in the US, we have about 1.35 million cases, 26,000 hospitalization and 420 deaths, and at least 400 million in medical cost. So this infection is a zoonosis, which means it can be transmitted from animal to humans, and is a foodborne infection, and in fact is primarily transmitted from food sources, although it can also be transmitted, for example, by pet animals. The typhoidal Salmonella is actually a smaller group of Salmonella, and these include the Salmonella typhi and Salmonella paratyphi A, B, and C. And this is a big problem worldwide. There is up to 27 million cases worldwide and 135,000 deaths. But as you can see, the infection is primarily localized in the Indian subcontinent as well as in Africa. And in the US, most cases are due to travel. They are imported from these areas. There are about 5,700 infections and 620 hospitalizations. And in contrast to the non-typhoidal Salmonella, there is no animal reservoir. So there is only humans are infected and can transmit. So the transmission is exclusively human to human. And some patients can become asymptomatic carrier, like for example, this is the case of Typhoid Mary, which she was a famous cook who transmitted Salmonella typhi to many people, and Salmonella typhi in this carrier colonizes the gallbladder. So what are the manifestations of disease in humans? Non-typhoidal Salmonella, they cause primarily inflammatory diarrhea. This is a disease characterized by fever and white blood cells in the stool samples. These are neutrophils. But in 5% of patients, children, elderly, immunocompromised, Salmonella can actually cause bacteremia. And typhoid fever is a different disease, and actually it manifests primarily with fever. In fact, in the endemic area, it's often difficult to distinguish typhoid fever from other diseases like malaria or brucellosis, which also are characterized by fever. In 30% of cases, patients get diarrhea, but the diarrhea is different from the inflammatory diarrhea caused from non-typhoidal Salmonella and is primarily characterized by mononuclear infiltrate. So what I'm going to tell you today, I'm going to cover some topics about the differences between the different Salmonella disease in humans, and I'm going to go over these topics in a little bit, and what we know and what we don't know. So first, let me introduce you to Salmonella in animal models, and the animal models that we can use to study inflammatory diarrhea as well as typhoid fever. So inflammatory diarrhea is caused by Salmonella typhimurium and other serovars that are broad host range. And that's why there could be a broad use of animal models that includes calf, rhesus macaques, and these develop a disease that is fairly similar to humans with diarrhea and neutrophil infiltrate. And macaques has also been used in co-infection studies, like for example with HIV and in malaria model. And the mouse model that is used is the streptomycin-treated mouse model, which does not develop diarrhea, develops some loose stools, with neutrophil infiltrate, and can also be used for co-infection study. And typhoid fever, and on the other hand, as I said, is only infectious in humans. So humans have been used, actually, human volunteers to study typhoid fever. And more recently, also humanized mouse model with humanized with human stem cells, and these mouse develop lethality and can be used to study the natural pathogens. But definitely one of the most used model is the mouse typhoid model, in which the mouse is infected with Salmonella typhimurium, which causes mouse typhoid. And in this mouse, there is lethality and monocytic infiltrate in the gut, and this mouse can be also used to study persistence and carriage. So many of these animal models, have been used to investigate inflammatory diarrhea and typhoid fever, and all these models have been useful to contribute to our understanding of disease. So what are the major virulence of Salmonella? So one major thing that distinguishes Salmonella, for example, from its close relative E. coli is the ability to invade the intestinal epithelium. And you can see here that host cell invasion occurs thanks to this virulence factor that is a type 3 secretion system encoded by the Salmonella pathogenicity island 1. This type 3 secretion system delivers effectors, and in this way mediates a Salmonella uptake by non-phagocytic cells. And SPY1 and its effectors are needed for the development of inflammatory diarrhea. So here you can see a calf intestine. So this calf was infected with wild-type Salmonella and developed this strong inflammation with neutrophil infiltrate. Calf instead that is infected with a mutant in this type 3 secretion system does not develop disease. And this type 3 secretion system encoded by SPY1 is not needed to spread beyond the gut. Salmonella encodes for a second type 3 secretion system encoded by the pathogenicity island 2, which also delivers effectors into cells, and it's necessary for Salmonella survival inside those cells and for the formation of the Salmonella containing vacuole. You can see here Salmonella replication inside the macrophages that is inside this vacuole, and this type 3 secretion system encoded by SPY2 also contributed to diarrhea, although to lesser extent than the SPY1 type 3 secretion system, and however, is primarily involved also in the spread of Salmonella beyond the gut. I'd like to mention also the SPV virulence plasmid. This has been widely studied here at UCSD by Don Gani and Josh Feeder, and some of the SPV encoded genes are actually effectors of SPY2. And this virulence plasmid is correlated with Salmonella dissemination in humans, so strains that have the virulence plasmid are capable to disseminate and cause bacteremia. So these two type 3 secretion systems encoded by SPY1 and SPY2, so these two pathogenicity islands, are really a key for Salmonella evolution. And this has been shown many years ago that acquisition of SPY1 was a primary factor of differentiation from E. coli to Salmonella. And then acquisition of SPY2 was a primary factor for Salmonella to evolve from the cold-blooded animal like reptiles to mammals. And you can see here, for example, the subspecies Enterica, which is the most prevalent one. And this now with more detailed genomic studies, we can actually evaluate the common ancestor and see how Salmonella bongorai and, for example, Salmonella typhi have evolved. And you can see that Salmonella bongorai has a limited number of pathogenicity islands and limited metabolisms compared, for example, to Salmonella typhi that has acquired a lot more factors to be able to replicate in the mammals and in the human host. So let's go over now the topics that I'm going to cover. So first, how does non-typhoidal Salmonella cause inflammatory diarrhea? So as I said, the Salmonella is an invasive pathogen, invades epithelial cells, can replicate in macrophages, and this triggers an inflammatory cascade with production of IL-23 and stimulation of the numerous T cells in the gut to release cytokines like IL-17 and IL-22, and the consequent production of CXC chemokines. Neutrophils come in, and neutrophils are able to kill Salmonella in the tissue. However, in the lumen, neutrophils can secrete also reactive oxygen and nitrogen species, which affects the gut microbiota. And at the same time, the production of antimicrobial peptides by neutrophils and epithelial cells results in a nutrient limitation. However, Salmonella is able to grow and replicate and acquire nutrients in this environment, which mediates the evasion of a host antimicrobial responses, fecal shedding, and transmission. So why do some patients develop bacteremia? So I can give you here the example of a project that I was involved with many years ago, in which we investigated the mechanism by which HIV infection enable and promoted Salmonella bacteremia. And so we used the simian immunodeficiency virus model in rhesus macaques, and we found that SIV in rhesus macaques promotes a depletion of Th17 cells, and this results in a blunted inflammatory response. So this response that I show you, this Th17 responses, is blunted in HIV-infected macaques, and this leads to increased bacterial dissemination and also, as shown by others, increased microbiota translocation. So now let's move on to the second group of Salmonella, the typhoidal Salmonella, and discuss why does typhoidal Salmonella not cause inflammatory diarrhea. So this has been puzzling for a long time because Salmonella typhi has the same virulence factors as Salmonella typhimurium, for example, and causes pathogenicity islands SPY1 and SPY2, although some effectors are pseudogenes. There is no virulence plasmid, however, which in the non-typhoidal Salmonella can promote dissemination in susceptible hosts. There is extensive genome degradation with a loss of function in over 200 pseudogenes in Salmonella typhi. And at the same time, though, there is a gain of function and acquisition, for example, of the SPY7, which encodes for the VI capsule, and also acquisition of the typhi toxin. So first, if we look at how Salmonella interact with host immunity, we and others found that the Salmonella typhi VI capsule is able to promote evasion of innate immunity. So Salmonella typhimurium for the host looks like this red noisy plane. So it triggers a strong inflammatory response, is able to activate TLR5 and node signaling and complement activation, and causes this massive inflammation that I showed you a few slides before. However, Salmonella typhi, by expressing the AVI antigen and by shutting down expression of the of SPY1 and flagellin with this regulator TVIA, is able to look like a stealth pathogen and basically fly below the radar and be largely undetected by the immune system. You can study this, for example, in animal models where you can express the VI locus in Salmonella typhimurium, and this causes reduced inflammation in comparison to what you see with the Salmonella typhimurium wild type. So this is a summary of what I just told you, that Salmonella typhi is able to fly below the radar, and the inflammatory response during typhoid fever in the gut is actually blunted. So there is much lower inflammation. We don't know why there is an increased monocytic infiltrate, but we just know that there is really no neutrophil infiltrate, and Salmonella typhi actually disseminates and is able to cause bacteremia. So, but still, how does typhoidal Salmonella causes typhoid fever? The short answer is that we don't completely understand it. However, there are some insights on important virulence factors that might contribute to some characteristic of typhoid fever. One of these factors is the typhoid toxin. So, Jorge Galan's group many years ago described that Salmonella typhi has this typhoid toxin composed by cytolethal distending toxin and PTLA and PTLA-B, which are homologous to pertussis toxin. This toxin has a peculiarity that is expressed only inside the host cells, and it causes cytoplasmic distension and cell cycle arrest. And what they show is that animal inoculated with a purified typhoid toxin develop some sign of acute typhoid fever, like stupor and leukopenia. And also here at UCSD, the Varkey group, in collaboration with the Galan group, found some important features of this typhoid toxin that it binds to sialylated glycans. And in particular, it binds to NEU5AC in humans. You can see here this strong binding, but it doesn't bind, for example, to NEU5GC, which is a very similar sugar in chimpanzees, which might explain why chimpanzees can be colonized with high doses of typhi, but they do not develop typhoid disease. Despite this terrific progress, however, we still don't know what is the role of the typhoid toxin in vivo. In fact, the recent studies in which human volunteers were infected with either Salmonella typhi wild type or a typhoid toxin deleted mutant actually showed that there was no difference in the development of the typhoid fever. And if anything, the patients infected with the typhoid toxin mutant develop bacteremia for a longer time. So perhaps the toxin plays a role in a more severe disease, which of course cannot be modeled in human volunteers, is too risky, or in chronic carriage. So future studies are really necessary to determine the role of the typhoid toxin in vivo. And to study also Salmonella typhi in vivo, investigators are now using, for example, humanized mice with human stem cells. And for example, in this study by Ferry Fang's group, a mutant library of Salmonella typhi was used to see what virulence factors were essential in this model, and it was found that the VI capsular antigen was essential, and so was iron acquisition by salmonella, and was also essential for lethality in this model. And in this model, they didn't find a role for the typhoid toxins, but and then again, these are mice, although they have a human immune system, so there could be some important factors that are not considered in this model. So last but not least, an important question is, why are typhoidal Salmonella restricted to humans? And we still don't know that. but at least we gained some insights of host restriction of Salmonella typhi by beautiful work that was done by my good friend Professor Stefania Spanò, both as a postdoctoral fellow and in an independent laboratory. And unfortunately, Stefania passed away last year, and she left us too soon, but this is kind of her legacy, so I want to bring her work to your attention. So Stefania basically found that Salmonella typhi is killing in mouse macrophages, and this is dependent of RAB32 and Block3. There is an unknown antimicrobial mechanism that is able to control Salmonella typhi replication in mouse macrophages. And Salmonella typhimurium, on the other hand, is able to evade this by secreting two effector proteins, GTGE and SOBD2, that are absent from Salmonella typhi. So this is an important mechanism of host restriction, although certainly there is more to know and to learn about why typhi is restricted to humans. So with that, I would like to thank you for your attention, and I would like to thank all the Salmonella researchers all over the world who have inspired this presentation and have contributed with their work to an understanding of this important and challenging human pathogen. I also like to acknowledge my funding sources. Thank you very much.